Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my honor and privilege today to welcome our speaker, Dr. Vince Calhoun from Georgia State University, with additional appointments at Georgia Tech and Emory University. Dr. Calhoun got his BS in electrical engineering from the University of Kansas in 1991, followed by a master's of engineering, biomedical engineering from Johns Hopkins University, a master of science in information systems from Johns Hopkins University, and then a PhD in electrical engineering from the University of Maryland in May of 2002. He's conducted primary research at Yale University and spent many years at the Mind Institute at the University of New Mexico. In 2019, he joined the faculty at Georgia State University, where he is the founding director of the Tri-Institutional Center for Translational Research in Neuroimaging and Data Science. This is a partnership involving Georgia State, Georgia Tech, and Emory universities, and which focuses on improving our understanding of the human brain using advanced analytic approaches with an emphasis on translational research, such as the development of predictive biomarkers for mental and neurological disorders. Dr. Calhoun has built a career on developing techniques for making sense of complex brain imaging data. The use of big data approaches and neuroinformatics tools to capture, manage, and analyze and share data is a major emphasis. Because each imaging modality has its limitations, the integration of these data is a vital need to understand the healthy brain, especially also the disorders of the brain. Dr. Calhoun creates algorithms which seek to map the dynamic networks of the brain structure and the involvement of genetics and how these are affected while being stimulated by various tasks or individual with mental illnesses. Dr. Calhoun is an author on more than 650 peer-reviewed journal articles and over 750 technical reports, abstracts, and conference proceedings. He served in many leadership roles in the field of neuroimaging and data science. He is a fellow of the Institute of Electrical and Electronic Engineers, past chair of the Organization for Human Brain Mapping, amongst other accolades. He's been awarded over $100 million and counting in federal research grants to study the topics of neuroimaging related research. And his talk today is entitled Brain-Based Biomarkers, a Focus on the Heterogeneous, Enhancing Sensitivity to Brain Disorders and Change. As always, we are streaming this lecture live and for recording via YouTube. And if you are watching on YouTube, thank you so much for joining us. Also, our specially selected 2020, 2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Calhoun via the chat feature in their Zoom sessions. I'll synthesize these questions and ask them on your behalf at the end of the lecture. And with that, welcome, Vince. We are so excited to hear about your talk today.
Thanks, Jack. It's a pleasure to be here. What I am going to be doing today is talking about some of our work in trying to move towards brain-based biomarkers, but in particular with a focus on heterogeneity, and how do we use that in order to enhance our sensitivity. So I'll go through kind of some preliminary work and motivation, and then I'll talk about sort of a number of different ways in which we've tried to tackle this issue. So I think when we're trying to develop neuroimaging biomarkers of brain disorders or health, there's often this sort of back and forth between a focus on sort of individual changes and canonical changes. Like, can we find something that's sort of representative, right, of a group or a set of individuals? And that's kind of a circle or a cycle. And if you look at the field, you see studies go back and forth between group differences, individual prediction, individualized change, then trying to develop more generalizable information. So I think this is really kind of an important cycle, and that's a little bit what I'll be touching on today. There have been sort of a huge increase in recent years in studies focused on trying to get at sort of prediction, right? Accuracy, classification, et cetera. This is a review from a number of years ago where we started to look at this, and you can see, sort of some of the accuracies here across various brain disorders. There's a pretty big spread. There's also, a trend of higher accuracies with smaller N data sets, which probably gets at some bias. But there's also, quite a bit of evidence that brain imaging can be useful and accurate at predicting various aspects that may potentially be important for clinical translation. And we can predict, not only doing sort of binary classification of, group A versus group B, but you can also look at sort of regression or predicting continuous measures like symptoms or cognitive scores, et cetera. And this is sort of a more recent review where we kind of looked at this. And a couple of the take homes we found here: when you incorporate multiple imaging modalities, you tend to get higher accuracies than unimodal. You can kind of see the distribution here. And the other is that, if you're looking at a patient group, the accuracy within the patient group tends to be higher than the control group. And this is probably because the emphasis in a lot of the studies were focused on sort of symptomatic aspects more relevant for patients. Another area that I've been interested in, and I think it's going to be really helpful as we try to, again, move towards biomarkers, are these sort of large-scale competitions. And so this is just an example of one we did just recently, which had a pretty good turnout. We had about 14,000 entries, and this was the sort of the first ever UK Biobank-based brain imaging competition, and it used functional MRI and structural MRI, almost 12,000 brains. And it was sort of a collaboration between TRENDS, the Human Brain Mapping Organization for Human Brain Mapping, IEEE, and the UK Biobank. And one of the reasons these are useful is because it's pretty easy for bias to creep in when you're trying to do classification and prediction, even if we really try hard to cross-validate, keep things completely separate. Competitions are a nice way to sort of try to benchmark and evaluate algorithms from groups that are completely outside of your own. Another area that I think is kind of an important area is sort of there's another cycle that I think is important here, which is sort of looking back and forth between data-driven approaches and model-based approaches. And I think there's a big area of need for more hybrid models that sort of blend those two. And what I mean by that is essentially using priors, right? So here's an example where, we've done quite a bit of ICA, of course. But one of the things about ICA is that, it's completely data-driven. So if you run it on a different set of subjects, you might find slightly different components. It might be a little bit hard to match, et cetera. You can kind of blend the best of both worlds if you use something called spatially constrained ICA. And so the way that we approach that is we took two fairly large data sets and we did sort of independent ICA analyses on resting fMRI data, identified the components that replicated between the two, and then we used those to derive a set of sort of component maps that we can use as priors in a spatially constrained ICA. And so there's a number of benefits here, one of which is you can directly compare across data sets very easily because the components are essentially linked. And you can see again here how similar these two connectivity matrices look. These are basically the cross correlation between the time courses associated with ICA spatial maps in two separate data sets, and they're correlated above 0.95. Actually, I think this is like 0.98 or something. And then importantly, if we look at group differences between schizophrenia and healthy controls here, you can see that these patterns also look highly similar. They're a bit noisy, they're small effects, but these are the ones we were really interested in. And if you threshold these maps, you're correlating at about 0.95. The other benefit here is that we can estimate this in individual subjects, right? So we're just taking the priors, we're estimating a result from one subject, so we don't need a group. And a third benefit is it basically provides sort of adaptive spatial maps and connectivity information that we can then compare and cross-validate quite easily. And these kinds of network-based approaches where we're looking at sort of maps as opposed to specific regions tend to outperform, region of interest-based approaches. Here's one example where we applied this to essentially predict a response to antidepressant or mood stabilizer in young adults who came into a mood disorders clinic, and a number of whom who were unable to be diagnosed because they were in the euthymic state. And some of them who were diagnosed incorrectly put on the wrong medication. And so working with Beth Osich in London, Ontario, basically scanned at baseline these individuals and then followed them up very carefully over a course of several years, and eventually determined which medication they were most responsive to that worked best for them. And we used the baseline image to predict that follow-up medication. And we were able to do this with an accuracy of about 95%. So that was quite promising, and it's something that could potentially be useful clinically, and especially given that some of these folks, when they're put on the wrong medication, can get ill. And it also hints at, we can identify networks that are most predictive in this case, and you can see the ones that came out shown here in color. And that can sort of hint a little bit at mechanism and potential, targets for future treatments as well. So again, it's just sort of an initial. And the other thing that I like about this study is it completely ignores the DSM diagnosis. It's really just looking at medication class response. Another example is shown here where we're looking at ECT response. And so this is a treatment for major depressive disorder. And some folks will respond and persist in their response to the treatment. Others will have an initial response and taper off. And if we look at predicting the accuracy post ECT with a baseline resting fMRI scan, we find about a 78% or 77% accuracy in predicting the change in the depression rating scale that was measured. Okay, so that's sort of where we want to head, and I think there's a lot of potential applications there that can be looked at. But there's an unsolved challenge here, which I didn't touch on yet in those previous results, which is we need to know what we're predicting in order to train a model to do the prediction, right? And so if we don't know that, then it's called unsupervised, right, or unsupervised clustering. And so here is an example where I've clustered data into 4 clusters, and you can always find groups of data that fall into clusters naturally. But if you take that data and then you map, say, in this case we had 4 mental disorders represented, each dot here is a subject, and we map the disorders onto the same representation, you can see it doesn't look particularly useful in terms of telling us anything about the diagnoses. And that's because the brain doesn't exist just to tell you about your mental disorder, right? There's a lot of things that the brain is doing, and the data is not necessarily going to be reflective of what you care about. And so this is one of the reasons it's such a big challenge. So one way we've tried to get at this is using approaches based on something called biclustering, and sort of subsequent extensions that we've developed. And this approach was initially developed to look at genetic data. And here is an example where you have conditions. There are H conditions here, and these are the genes and their responses, whether it was sort of on or off, positive or negative, or non-responsive. And what biclustering is doing is to jointly group conditions and genes together. So here you can see, data like this gets grouped into 2 different biclusters where genes 1, 4, and 9 show consistent on-off responses to the different conditions there. Genes 1, 4, 6, 7, and 9 show consistently positive responses to conditions B, E, and F. And so you can think of this as, so you can kind of get at here looking at subgroups of individuals, right? Or potentially subgroups of responses or brain patterns. And so that's what led us to sort of our initial study looking at structural MRI. And so what we're doing in this case is we first took some T1 structural MRI data, segmented it, and then applied ICA to those maps, something called source-based morphometry. So it's like a multivariate extension of VBM, or voxel-based morphometry. We're essentially extracting maximally spatially independent patterns of gray matter that covary in a similar way among subjects. And so what we decided to do was to take these loading parameters, which tell you the degree to which a given subject expresses a given source map, and do biclustering on that. And so we, with the biclustering algorithm, we're essentially taking two components here, and we're jointly clustering subjects and components together. And this was in the case of looking at symptoms in individuals with schizophrenia and a healthy group as well. And so when we're looking at symptom subtypes, we essentially found three sets of subjects or subtypes, one of which was heavily weighted on positive symptoms and loaded on one component here. Another one of which was heavily loaded on negative symptoms and loaded on a different component. And a third one, which was sort of jointly loaded on both components and was more generalized in terms of associated with generalized symptoms. And so you can kind of see where this is going. This is really trying to get at, can we look at the variability in our data and see where we have consistent sort of apple-to-apple comparisons. And so another example where this, which sort of highlights the benefit of this type of an approach, is if we extend this to sort of n-way clustering. So instead of just two, we have multiple. Same kind of scenario where we have structural MRI and we have loading parameters here, but we bicluster on all the combinations of the components. And we also include symptoms, right? And so we have positive, negative, and generalized symptom categories that are also included as data types there. And so what we found was pretty interesting, I thought. So first we have biclusters here that only include components, and you can see component numbers listed here. And I know that doesn't mean a lot. The specific numbers are shown here. After the semicolon tells you how many of the subjects were grouped into that bicluster. And interestingly, you can see here where the black circles are that we get significant relationship with various symptom categories, which does not is not reflective if we don't do the biclustering. We don't find a strong relationship with symptoms. And the reason for that is because what we're doing is we're sort of focusing on homogeneous subsets of individuals. Again, more of an apples to apples comparison. And within that, the variability is lower and the relationship to symptoms emerges. And likewise, we find certain patterns of symptoms that are linked to certain components, right? So here we have a component 14 is covarying, these 60 subjects covary between component 14, positive, and generalized symptoms, okay? So this is a lot of data. There's a lot to take in here, but I think it's a really useful way to start to tease apart some of these data sets while getting at heterogeneity in individuals. And of course, we kind of keep going here. So we said, well, can we apply this to fMRI data and look at dynamics? And so we did that, where essentially we're jointly clustering on windows, which is basically sliding windowed cross correlation patterns you can see shown here, right? Sets of subjects and pairs, meaning individual cells within these connectivity matrices. Okay, so here we've got, what you're doing in dynamic connectivity is you're essentially computing the functional connectivity in sort of a sliding windowed fashion, and then you can cluster those into states, patterns, transient patterns of connectivity that individuals move close to or farther away from over time. And so with the tri-clustering approach, we basically applied it to a large data set of about 300 plus individuals with schizophrenia and healthy controls. And we're jointly clustering, as I mentioned, windows and subjects and pairs. And so what we found was, again, an analysis that showed a much increased sensitivity to patients versus control differences. And here you can see if a cell is white. So these are 5 different states that occur at different times in different individuals. These are, this is the control, the patient, and the difference. And all of the ones that are shown here are significant. The ones that are white are excluded from the tricluster because they weren't homogeneously associated with that set of, again, here you can see 3,500 windows, 81 subjects, and 619 pairs in this matrix that came out. And we found 3 of these were individually linked to either generalized, negative, or positive symptoms. And there were actually quite a lot of interesting results that are fleshed out more in this paper that I've listed here below. Okay. Another source of variability that we've been trying to get at is spatial and temporal variability. Okay, so the dynamic functional network connectivity tries to get at that to a degree, but you know, the majority of temporal dynamic studies have focused on just coupling between fixed regions, right? So the correlation between these two changes over time, right? It goes up or it goes down. But you can also imagine spatial dynamics where maybe the nodes that you're looking at shrink, or maybe they grow, or maybe they shift a little bit. And then you can have joint spatial temporal dynamics, right? So I think these latter two are only just beginning to be studied in the field. There's been quite a few studies of temporal dynamic in the field. And what we're finding is that there's actually a lot of evidence that spatial variability in these functional patterns that we're seeing. This is just sort of one initial way to kind of look at that within each. It's sort of a hierarchical analysis. So within each sort of domain, we can look at sort of subclusters and how they couple differently over time, create little transient movies within the domain. So this is, for example, subcortical network domain, and you can see how it changes over time. And importantly, if we look at a patient and control group, we see the coupling varies quite substantially between the two over time in just the spatial aspect of the dynamics. Okay. And then a third category where we're kind of getting at that as well is, well, instead of just focusing on a pattern of connectivity that, or fixed states, right, that occur. Maybe we have 5 states. They occur at random times in different individuals if it's resting data. But maybe those states actually have some behavior that occurs smoothly over time. Can we look for sort of motifs or transient changes in these larger state patterns? And so those we call statelets, essentially sort of short little transient motifs around those states. And interestingly, well, why did we do this? Well, when we started to look at this in a patient data set, we find very striking differences between the two. For example, the healthy individuals are showing much more evidence of longer recurrence, right, over time before it kind of tapers off and disappears, almost like a ripple in the water. Whereas the patients show more sort of an up and then almost off fairly soon. You can see here the time decay. It's almost night and day, right? We see a fairly dense link here when we look at time decay in the healthy individuals, whereas there's almost very little to nothing in the schizophrenia patients. Classification, using time delay is significantly higher than it is when we're using something like connectivity patterns, for example. So again, this is just another example where, again, trying to dig into the data and represent it in a way that captures some of that variability. Okay, the other sort of elephant in the room is always the diagnostic labels, right? So, for example, if we're talking about schizophrenia and schizoaffective disorder, some of which are individuals that experience manic episodes, others more depressive. Those are all based on self-reported symptoms and interviews, and they're not biologically based, right? They're useful clinically, but they're not biologically based, so they're not really getting at the core of the disorder. And so we know that they're not perfect, right? And so how do we deal with that when we're trying to do a classification study? Here's an example where we just applied a technique trying to look at labels. So in this case, a label would be a diagnosis. And we treat those labels as noisy, right? So we allow them to have some error, right? And you can do this. One way to sort of get at, well, is there error or not, is if you apply multiple classifiers across, across the data, and then you look at whether the classifiers agree or whether they disagree consistently. And so that gives you some evidence of whether they're noisy or difficult or challenging, and should we relabel them potentially. And you can do this. We applied this to this approach to some numeric data. So this is the MNIST data. And we took numbers that sort of look fairly similar and kind of could be confused with one another. So 8, 3s, 5s, here's a 1. And you can see if we apply this approach to basically identify the noisy labels and relabel them, we improve the accuracy up to a certain point. So it doesn't overestimate, it actually is improving the result in this sort of well-defined case. And then we decided to apply this to structural imaging data from this psychosis spectrum study. And we are looking at it with sort of 2 different categories. This is the BSNP study, which is looking at trying to define something called a biotype, which is using a set of data that includes biological data and trying to identify sort of a clinical type, right? And sort of ignoring the diagnosis, right? So they have in that study identified 3 sort of biotypes. In this case, they're labeled 1, 2, and 3. And we also have the DSM categories, in this case, bipolar, schizoaffective with psychosis, et cetera. And when we look at sort of original versus cleansed, which is sort of denoised, you can see that the accuracy increases, right, significantly. It goes up quite a bit for these pairwise comparisons, as well as for the all-way comparison. But importantly, we've got, again, in this case, 2 different categorization approaches. One is DSM, one is biotype. And if we apply either one of those, we see an improvement, and we see sort of meaningful separation. And when we look at the voxel-wise comparisons of the relabeled data, the regions that come out as showing differences in gray matter are sort of meaningful, right? And they reflect some of what we see in previous work, and much more sensitivity than what we typically find. But they're not the same, right? So this kind of then gets at, well, what else could we do here? It's almost like using a prior, right? You're taking your DSM labels as a prior, but it's not fixed and it can shift, right? If you take biotype, you might get a slightly different answer. So we can think about, well, maybe we should use multiple categories, use all the information we have at hand, or you can get there's a lot of things this sort of could lead to, but it's really trying to say, well, let's just expect that the DSM categories are error prone and not biological, and allow the algorithm to try to update those. Okay, and then the last topic I wanted to touch on was, sort of heterogeneity in the context of deep learning. So deep learning has had sort of quite a lot. Let's see if I can turn off my menu here. Hide video panel. Try that. Whoops, no. Hide floating meeting controls. Okay, I'm never going to be able to get those controls back, but oh well. So here was sort of so we've been looking at deep learning for, a number of years now. And it's clearly beneficial. It clearly has some areas where it can give you information that you don't get from standard approaches. Here is one more recent study where we looked at some large N data. We used the UK Biobank data, and we looked at comparing deep learning with what we're calling standard machine learning. And so that would be things like linear discriminant analysis, logistic regression, support vector machine. We divided the categories into groups of age. So there were essentially 5 different age groups, as well as females versus males. And you could see in all cases, these top 2 black and blue bars on this graph are the 2 deep learning approaches. And these are all the standard machine learning approaches. And you can see even up to 10,000 subjects and even down to, only 100, where deep learning is outperforming in terms of just the accuracy, right? But of course, we don't want to treat this as a black box. And one of the strengths, even though it's talked about often as a weakness, deep learning, is giving you access to a bunch of parameters, right? And those parameters, the challenge is how do we visualize those? But we have access to them, so there are lots of strategies for trying to visualize them. And so, for example, in this case, we looked at Alzheimer's versus mild cognitive impairment, stable and progressive versus controls, and you can one way you can do this is something where you sort of visualize the layer and you project it back into brain space, and you can see, for example, medial temporal regions are the most informative when we're predicting Alzheimer's versus controls, which makes sense, based on prior work. We find about a 93% accuracy there, but more importantly, for stable versus progressive MCI, predicting future decline, we're getting above 80% accuracy. But it is important to kind of go back and visualize. Another way you can visualize is to use these sort of projections, right? So this is using something called T-distributed stochastic nonlinear embedding, or TSNE. You basically take a bunch of parameters and you project them into a 2-dimensional space, which preserves the local, neighborhood information. So each dot here is a subject, and this is just showing that if we go from 1 to 2 to 3 layers, which is sort of small for deep learning models these days, you start to see some interesting clusters here come out for these groups. This schizophrenia, bipolar disorder, schizoaffective disorder, and it gives you some interesting observations. And we can also then drill down into each individual subject and visualize, how they look relative to the others. But we're just getting started, right? So here's another example. Let me again hide the floating meeting controls. Okay. Here's another example where we're asking the question, is our model complexity optimal, right? And so using resting fMRI here, we're comparing, components in different domains here and how they are linked, or functionally connected to other component domains. And whether that is that sort of accuracy when we're trying to predict the clinical group is improved if we use sort of a linear relationship versus an additional layer versus a third layer after that. And what we're finding is, and these are all cross-validated, what we're finding is evidence that, for example, you can see here the cognitive control networks show evidence of needing additional layers, additional complexity. Their relationship is there, but it's best modeled. It needs a more complicated model, more parameters in order to model it to achieve the highest accuracy. So again, you can see this is really, there's a whole bunch of things we can do here, right? But it's really getting at, can we use this information, the structure of the model that emerges from this, to tell us something intuitive about brain regions and brain domains. And then I think the last example here is using deep models to try to get at the temporal information a little bit more. So you can use something called convolutional neural networks, lets you leverage the spatial information. Recurrent neural networks let you leverage delays in time. And so if you combine the two, for resting fMRI data, you can develop a model that predicts from resting fMRI or from other kinds of fMRI data or temporal, spatial temporal data. And just as I had mentioned before, we can project down into these two-dimensional plots here and see where the patients and the controls lie relative to one another in both the training and the testing data. We can visualize which components, in this case, we fed in to the model. We didn't use raw data in this case. We're actually feeding in ICA components. And we have individual components that are most discriminative in this particular model. And we can look at the relationship to other variables of interest, for example, symptom categories. And some interesting things emerge. So for example, if we look at healthy and schizophrenia, we can see that the symptom scores kind of have this gradient where you have more severe and least severe, and the least severe are closer to the controls. Again, the model here knew nothing about symptoms. We're just mapping those symptoms onto these projections, post hoc. And this is the last one, I promise. So here we're looking at a four-way classification using, again, a deep learning model. When we apply the projection, we found this interesting result here where the schizoaffective group was split into two different clusters, right? One that was closer to the other groups, and one was kind of off on its own. And you can see that the symptom scores don't help you a whole lot. There's evidence that maybe there's a pretty big spread here in this group as there are in that group. But these kinds of visualizations, and we're kind of working currently on some different ways to do this, but trying to visualize, well, what is unique about those folks that are misclassified or folks that are sort of well classified? And can we look at that in some sort of a continuous type of measure? So in summary, there's a lot of information that we've mostly been ignoring that we can obtain when we focus on subgroups of data or subspaces within our data. If we focus on this sort of homogeneity and heterogeneity aspect, it can really enhance our sensitivity to relevant information, for example, severity of illness. And as the data sets are growing, and more open data sets are available now, this gives us an opportunity to help us try to advance our ability to characterize mental illness and replicate these results. So, just a slide here to mention that we try to release our methods. We've got a number of toolboxes that release that implement the approaches that I have mentioned in the previous slides. And we, like many others, are a big proponents of data sharing and also data capture in sort of systematic ways. And so we have a system called COINS, which sort of all-inclusive does imaging and assessments and radiology reviews. But an interesting piece of that is sort of complement to that is decentralized analysis. So with these sort of large studies, if you want to do an analysis, you have to have a bunch of infrastructure and download everything, or you have to have a cloud account or access that account. And if you want to combine it with a data set from somewhere else, you have to get them in the same place or find a way to do them separately. Even more so if you want to work with groups like Enigma, where about half of their data sets cannot be centrally aggregated. You can't analyze them with more complicated type algorithms unless you have approaches that can, do analysis in sort of a decentralized way by passing around metrics from data set to data set. We have a tool we built called coinstack, which is open source and does this sort of analysis. And so that's kind of an aside. If you're interested, you can check it out here. The work in that I presented today is basically, I just have to thank the folks at the Trend Center, many collaborators that we work with, and our funding sources, and also thank you very much for bearing with me. I think I'm about on time, and without further ado, I guess Jack will take some questions now.
Oh,
Me to unshare my screen.
You can just unshare your screen so we can see you. That would be just fine. Vince,
Okay.
firstly, thank you so much for kind of going through, just really a remarkable amount of work from you and your team and from all your collaborators. I mean, it's really quite an impressive amount of firepower you guys bring to all this. I was, as we're kind of waiting for a few more people to fire off some questions, I was very interested in about finding out a little bit more about the spatially constrained ICA. I suppose I've always been a little bit concerned about ICA's ability to on convergence and comparability across subjects, and whether or not the ICA analytic solutions are mathematically unique. And it sounds like spatially constrained ICA may help with that, and maybe get a little more network consistency across groups. I was just kind of curious if that was one of the motivating factors behind the spatially constrained ICA, or if I'm off base here.
Yeah, I mean that is the ability to kind of run this on individual subjects and have comparability across components. I mean the group ICA framework that we've been using for years is kind of a way to get at that too. Basically what you're doing is saying, I'm interested in comparing data sets, and therefore I'm imposing upfront this group model, right? And this is kind of the reverse of that, which is, in the sort of standard group ICA approach, you are running a group map and then you're reconstructing individual and comparable sets of components from individual subjects, right? So within that data set, things are comparable and things work well, and we can get the same answer if we run it multiple times, assuming we do these we do these repeat analyses to kind of guarantee that. But if you want to cross-validate or analyze data outside of that initial data set, up till now have had to basically resort to running another ICA and then figuring out kind of how it matches, and it may not.
Right.
Or just throwing away the ICA model and just going to like a regression-based approach. And so we were interested in sort of leveraging the benefits of ICA and maximizing independence and having all those guarantees, but also allowing us to do this outside of that original data set. And so that's kind of where it led us in that direction. And so we've been running, all sorts of analyses now with that. And the other thing that's nice is you can do a bunch of stuff within your study, but if you do another study and do something completely different, you can compare, right, across those studies if you use those techniques. So it's quite nice. I like it a lot, but you guys can try it out. We have the maps up there on our website, and there's a paper which I think it has Neuromark in the name. It talks about it. And so feel free to try, and it's built into the GIF tools as well. You can just click on it and run it on one subject if you want.
What were the, you've had the benefit of having some pretty remarkable sample sizes to be able to put through some of your methods, especially with some of the, I'm sure this is important not only for ICA, but also some of the bi-clustering stuff you talked about. There are several of those big data sets like the Human Connectome Project or ABCD for development. You mentioned the UK Biobank, I think, somewhere in there. What sort of sample sizes do we kind of need to have? I mean, I'm thinking the UK Biobank is just a ridiculous amount of data, but obviously that's kind of a rare resource. But what sort of sample sizes do you guys look for when you're trying to run ICA kind of analyses?
I mean, in terms of what you need, well, I think it really depends on what questions you're asking.
Okay.
And I don't think there's a minimum per se. I would say if you're interested in individual prediction, sort of accuracy scores in a binary classification case, then, more is better, right? Because you can, there's a certain variance that you have to account for around your accuracy, and you're kind of bounded by that. If you say you have, 100 subjects, and your variance is 5%, right? Plus or minus 5% or plus or minus 10%. Well, that can be a pretty big difference, right? If you're talking about guiding clinical decisions, right? And so the more you can ramp up that data set, the more you can reduce that. and be more confident in the answer. But so I guess I would say there, but that doesn't mean that you're not learning some useful information from the smaller data set. So I guess I think it really does depend on what you're predicting and how large that effect is in the data. It's why we've kind of tried to look at group effects and do they replicate, things like that. So
yeah, I was going to say that one of the
but I'm not, and again, like if you're predicting behavior and behavior is highly variable, then that's gonna be your rate limiting step, right? Your rate limiting, part of that study is the fact that your correlation's like, on average 0.2, right, or 0.3.
Yeah,
that's going to be hard to predict, right, with any level of accuracy. So.
On your spatiotemporal dynamics application, I was particularly interested in that. And I was just kind of curious if you've noticed how the variability spatially is structured. Like, for example, is it cytoarchitectonically constrained? Are you notice any things that like that? And also, does the time scale play an important role of the data acquisition?
Yeah, so I'm pretty sure there will be some interesting cytoarchitectonic links there, but we haven't tackled that per se. Just, I would do it if we had a 7T scanner and try to get it, so I think the layer specific stuff can help a little bit with that. It doesn't get you cytoarchitectonics, but it gets you a little bit of input output, right? And things like that.
Right.
So we've been looking more at sort of the macro scale, right? Because we're kind of working with more standard 3T data sets at this point. I think the time scale does matter. And you see things that are kind of, as I showed in that statelet analysis, the drop off of the, hanging together of the state is much faster, right, in the patients than it is in the controls. And there's a lot of interesting elements that you can glean from delay as well. I think ultimately we also have to remember that we're looking at, again, very macroscopic level data, and I didn't really get into sort of multimodal, but you can do multimodal analysis and collect multiple types of data from the same person. And then if you have a number of subjects with those same data types, you can start to try to link those scales together. And that can give you more information as well. And I think there's a lot you can do there.
That's great. Are you aware of any efforts to apply models like the ones you've described to patients that do or do not respond to therapies?
I'm sure that yes, I've certainly come across some of those. So the idea would be like selecting individuals who would be most responsive, right? So that would be one potential scenario where you could use it. We've talked a lot about just in the context of not necessarily therapy, but treatment in general. Can you pre-select individuals who are more likely to be responsive, right, to a given treatment?
Right.
And so, but yeah, I mean, I think that would be, I haven't done that myself, looking at sort of response to therapy. I think there's some data where at the Mind Research Network, Eric Klaus and Katie Witkowitz are looking at sort of cognitive behavioral therapy and mindfulness and some other measures like that. And so that will be eventually something where we can try that on. But yeah. I think that's a great area to apply this to.
That's great. Now, one of the things which is always seems to be you mentioned rate limiting factors is that individuals don't often have necessarily access to high performance compute clusters at their institutions for whatever reason, or maybe they're not really fully bought into giving money to Amazon. I was curious if you might tell us a little bit more about coinstack.
Sure. Yeah. So what we're doing there is, and we call it decentralized learning. I think Google calls it federated learning. Essentially, what we're doing is you download this application and you map your data to it. So you maybe map your structural MRI data to it. And then you see consortia and you can join a consortia, right? So suppose you and 5 of your friends download coinstack and somebody creates a consortia, and you join that, right? And then the consortia creator sets up an analytic model or a computation, and that might be do a VBM analysis of these data, or it could be a free surfer analysis, or it could be, we have group ICA in there as well, this sort of spatially constrained group ICA. And then, and then what happens is that they specify which variables are needed for the analysis, and then the individual sites see those variables, and they map their, do the mapping of their variables to those, right? So the names match. They don't have to match, but you match the data.
Right.
And then you click go, and then it runs the analysis locally. So basically, it takes that analysis and it runs locally without moving the data. It produces some derivatives, potentially adds noise to that, and then aggregates it privately and can iterate. So you can, you may need just one iteration, you may need many, depending on what you're doing. If it's just a regression, one will be enough. If it's like a support vector machine, you need more. And then you get a global, answer to that solution. And so that again, has multiple benefits, and it's complementary. If you can put all your data in one place and have everything and have access to all of it and run it there, that would be great. But sometimes you don't have the resources. Sometimes the data can't be shared, right? So you're collaborating with somebody and they're not allowed to. We have a data set, this FBurn data set that I've been showing a lot here. We've been arguing with the IRB for over a decade, trying to get them to allow us to share it, and they will not let us share it.
That's amazing.
And because it didn't have data sharing language in there when it was collected, which was way before this became common.
Yeah.
And so there may be cases where you have rare disorders, or you have identifiable information, other kinds of data that you're looking at that might be sensitive. And so I think it's a useful complement, and you can imagine connecting that to large pooled data as well. So pool what you can and bring in what you can, right?
Right. Are there any hardware requirements that if someone wanted to become a coinstack a node in your system, would they have any
it's at the moment it's just run on individual analyses, and so it really depends on what you want to run.
Okay.
So the rate limiting step is how much local data you're analyzing. So if you're analyzing a lot of data locally, you need to have enough storage for that data and enough computation to run it.
So it's whatever your local scale happens to be, right?
Yeah, right, exactly. So, but you don't need to scale it to the entire amount of data that you're analyzing across all the sites. The other thing we've got, we're sort of working on this now. We've got it sort of a pre-alpha version of it. Is like a sort of a headless coinstack, I guess I'll call it, which is if I have a a data set and I just want to make it open, right? For coinstack people. So you can't get the data, but you can always see the coinstack node, and you can control the mapping, right? And run the analysis. Then that would be a great for some of these larger data sets where you want to provide a resource, right, for the community, or a benchmark for people, or et cetera. And so that I think is something that could be hosted, and potentially should require maybe a data center if you're going to have a lot of people using it, right?
Right. Are there any? Is it all or nothing? Either you're only say sharing like statistical results or the raw data, or not at all, or are there kind of levels of granularity of what you can be shared?
So yeah, it depends again on the computation you do. So there's a couple levels. Like you can make consortia private if you want. So only your friends can join. You can pick an algorithm that sort of passes derivatives, like maybe it passes gradient weights, or maybe it passes beta values or something like that, right? You can also do something called differential privacy, which it takes the derivative, and maybe you feel like I don't even want people that, I'm worried that people might machine learn on my data, right, and figure out, even though it's not that informative, they might still be able to put those pieces together, right? And so differential privacy allows you to add some noise, structured noise to it that then gets sort of averaged out when you do the aggregation. And we even have another one that's using encryption now, which is basically sort of bank level encryption, where you encrypt the derivatives before you share them, and then you learn on the encrypted derivatives. So there's different levels there that we're working towards, for sure, because different people will have different needs. So,
what is next in the world of big data, brain science in this space, do you think?
I don't know. There's so much to do here. I mean, I feel like I think we're learning a lot, but I guess the things that I worry about, I guess I'll say, are there's too much turning of the crank without thinking about what's underneath the crank, right? And so, for example, I like the fact, I mean, I love all this. I love the fact that there's a lot of data available, but I'm also wary that, there's a lot of reuse, and studies are going to start to be correlated to one another, right?
Yeah.
And another thing is that if everybody uses the same pipeline, right, to process their data, you've baked in biases, right? So think about biological diversity, right? Analytical diversity is probably good too. It's not necessarily a bad thing. And it can give you some benefits. It can give you, some information, right? I do think that that there, things are accelerating, and I feel hopeful about having brain imaging lead towards some more useful clinical tools, before too long, because I feel like we're sort of getting a better handle on, replicability and generalizability and things like that. Still working on that, but these large studies are helping.
That's great. Well,
and then, yeah, and I'm still we're doing a lot with deep learning.
Yeah.
And I think, again, developing models that are interpretable. So one of the ways that we're doing that is we're taking ICA, which one of the things I like about it is I find it somewhat interpretable to get out, multiple networks that interact, with one another. And so you can build that inside of a deep learning model, right? And then start to look at, instead of just this sort of massive set of hundreds of thousands of weights, you now have them segregated by networks. You can look at subspaces. That was kind of what I was getting at with the complexity analysis I showed. Right. So anyway, there's just a few things. Someone else will give you a completely different answer, probably.
That is undoubtedly the case. Well, Vince Calhoun, thank you so much for joining us and sharing your work with us. What an amazing amount of work, and what an amazing team you have. And I hope everything is going well for you after the big move to Georgia. But anyway, thank you everybody who joined us, and we look forward to seeing you next week. Thanks again, Vince. Appreciate it.
My pleasure. It's a pleasure to be here.